So welcome to part 6 of the muscular system. This is actually our final video on the muscular system, and we're just going to spend a little bit of time looking at smooth muscle. Smooth muscle was introduced during part 1 of the muscular system, but just as a bit of a recap, remember that it is found in walls of most hollow organs, except of course the heart, which is cardiac muscle. It consists of spindle-shaped fibres, which are thin and short compared with skeletal muscle fibres. It only has one nucleus and no striations. It lacks connective tissue sheaths and has the endomysium only. The sarcoplasmic reticulum is less developed than it is in skeletal muscle, and it has pouch-like infoldings of the sarcolemma called caveolae, which hold and sequester the calcium. However, most calcium influx actually occurs from the outside of the cell, and this is a very rapid process. It also contains no sarcomeres, no myofibrils or T-tubules. Usually we find smooth muscle in two layers. Firstly, a longitudinal layer. The fibers are parallel to the long axis of the organ. When they contract, they dilate and shorten. We also have an inner circular layer where the fibers are in the circumference of the organ. So when they contract, we have constriction of the lumen and it tends to elongate the organ. This arrangement allows for peristalsis. This is the alternating contraction and relaxation of the smooth muscle layers that helps to mix and squeeze substances through the lumen of hollow organs, for example, our digestive system and our urinary system. It is also interesting to note that there are no neuromuscular junctions as there are in skeletal muscle. Autonomic nerve fibers innervate smooth muscle at what are referred to as diffuse junctions. Instead of neuromuscular junctions, we have structures called varicosities or bulbous swellings of the nerve fibers, which store and release the neurotransmitter into those diffuse junctions. There are also a couple of other differences. First of all, the ratio of thick to thin filaments is much lower in smooth muscle compared to skeletal. In smooth muscle, there is one thick filament for 13 thin filaments, compared to skeletal, which has one myosin filament to 2 actin filaments. We also have structures called dense bodies, which anchor the actin to the sarcolemma at regular intervals. The myofilaments are spirally arranged, causing the smooth muscle to contract in a corkscrew manner, with the thick filaments having heads along the entire length of the muscle. However, actin and myosin still interact by the sliding filament theory. There is no troponin complex. Instead, we have a protein called calmodulin, which binds calcium. The cells are electrically coupled by gap junctions, which allows ion passage from cell to cell to help synchronize their electrical activity and results in slow synchronized contractions. Action potentials are transmitted from fiber to fiber. Some cells are self-excitatory, meaning that they can depolarize without external stimuli, and they act as a pacemaker for these sheets of muscle. The rate and intensity of contraction may be modified by both neural and chemical stimuli. But as with skeletal muscle, the final trigger is an increase in intracellular calcium. This calcium is obtained from the sarcoplasmic reticulum as well as the extracellular space. So, after there is a signal, the calcium ions either enter from the extracellular fluid via calcium channels or from the limited sarcoplasmic reticulum. The calcium then binds to the calmodulin. The activated calmodulin activates the kinase enzyme, and this kinase enzyme is called myosin light chain kinase, or MLCK. The activated kinase enzyme catalyzes the transfer of phosphate to myosin, activating the myosin ATPase. The activated myosin forms a cross bridge with the actin of the thin filaments, and we will have an increased muscle tension. And again, as with skeletal, if it is greater, the tension generated is greater than the opposing force, we will have shortening occurring. So when we have contraction of smooth muscle, they tend to be very slow to contract and relax, but are able to maintain it for prolonged periods with very little energy cost. It has very slow ATPase, and the myofilaments may actually latch together to save energy. The relaxation of smooth muscle requires calcium detachment from the calmodulin, very similar to removing the calcium from the troponin complex. We then actively transport the calcium back into either the sarcoplasmic reticulum or the extracellular fluid. And we have the dephosphorylation of myosin to reduce the myosin ATPase activity, same as skeletal. So we can have regulation of contraction of our smooth muscle by nerves, hormones, or local chemical changes. The neural regulation involves neurotransmitter binding, which increases the calcium in the sarcoplasm, and these could be either graded or local potentials or action potentials. The response depends on the neurotransmitter. For example, acetylcholine, adrenaline, or noradrenaline may be released, each resulting in a different response, and it also depends on the type of receptor molecules that are available. We also can control smooth muscle contraction via hormones and local chemicals. So some smooth muscle cells have no nerve supply, but they will depolarize spontaneously or in response to a chemical stimuli. Some respond to both neural and chemical stimuli, and chemical factors obviously include hormones, carbon dioxide levels, and pH. There are also a couple of special features associated with smooth muscle contraction. The first is the stress relaxation response. This is when the organ responds to stretch only briefly, then adapts to the new length. It retains its ability to contract on demand, but enables organs such as the stomach and bladder to temporarily store contents, and so therefore can expand and remain at that expanded position for a little bit longer. It also has length and tension changes, so these muscles can contract when between half its length and twice its resting length. So not as finicky as skeletal muscle. They can also undergo hyperplasia, in that smooth muscle cells can divide and increase in numbers. For example, estrogen has a significant effect on the uterus at puberty and during pregnancy. And lastly, this is just a comparison between smooth versus skeletal muscle in how they actually contract. There are very similar processes, but there are a few differences that you can see in the pink.